Okay, okay, it's done now. You can share your presentation.
Okay, presentation, share screen. Okay. Firstly, I have to share my video.
Uh, you can start your video and with the present, you can start your presentation both simultaneously. Okay, so we can good to go now. You can share your presentation.
Okay. Uh, is this visible to you?
Yes, yes, it is visible. Okay, so everybody, good morning again in the next session of our certificate course on regulatory and forensic aspects of herbal formulation. We are here with the madam, Dr. Shruti Bhavsar. Madam has done her BSc with the zoology in 2010 from the MG Science Institute, Ahmedabad. She has completed her master's in 2012 from the School of Science, Gujarat University, Ahmedabad. She has received her professional education degree B.Ed in the 2013 from the Gujarat University. With the entrance exam, she got admission in the PhD directly after MSc, and she has done her PhD in 2021 with the zoology as the main subject, cell biology, a specialized subject. Madam has worked as in cytogenetics and pharmacognostical parameters. It she has attempted the safety analysis of Calancho pinnata medicinal plants and its genotoxic effect at the higher doses. Her research work has been attempted for the very first time worldwide. She has published three research papers as the main author in the national and international reputed journals. Madam has also presented two posters in the international and world conference. She has attended more than six national and international seminar, five conference, five workshop, and one training program during the tenure of her PhD. She has also attained the nature education camp organized by GER Foundation Gandhinagar two times as the lead faculty. Currently, she is working as an assistant teacher in the Radiance School at Sargassam, Gujarat, Gandhinagar. Madam has also received Young Researcher Award in the 2022 for her one of the publication from the INSC, that is the Institute of Scholar. She is also recognized as a reviewer of INSC international publishing platforms and for the technical events under the stream, Basic and Medical Sciences. She has been recognized as a professional member of INSC International Publication Publisher in the recognition of professional standing. So, with this brief introduction, I welcome you, ma'am, for this as a resource person into the certificate course. So, over to you, ma'am. You can proceed with your session.
Okay. Thank you, Kushbu. Uh,
Ma'am, you have to share the screen again because there will be a black colored only we can able to see
okay.
instead of presentation.
Okay.
So,
Okay. I'm sharing again. You
now your screen is visible, ma'am.
Yeah, Kushbu ma'am, is this visible now?
Yes, it is visible and you are clearly audible, so we can start.
Okay, so let's start. Um, Hello everyone. Um, your topic that is Introduction to Genomics. I am going to share some information regarding genomics to you. As Khushbu ma'am told that genomics and genetics, these topics are very new to you. So I am going to conduct some basic information only. Uh, okay. So firstly, what is genome? That is regarding genomics. So according to NIH, that is National Human Genome Research Institute, genomics is a study of all of the person's or plant's genes. That is the genome, which includes interaction of those genes with each other and with the persons or plants environments. So if we are talking about genomics, it is the study of whole genome. Like each and every cell has genome and it cover all the genome, right? So what is the genome? Genome is a fancy word of all your DNA. I hope you all know what is DNA. From potatoes to puppies, all living organisms have their own genome. Each genome contains the information needed to build and maintain that organism throughout the life. So, your genome is the operating manual containing all the instructions that helped you develop a single cell into the person you are today. You, your features, everything is depends on your genome. So it guides your growth, helps your organs to do their jobs, and repairs itself when it becomes damaged. And it's unique to you. Unique means each and every person has their unique genome. That's why all are different. The more you know about your genome and how it works, it the more you will understand your own health and make informed health decisions. Okay, so this topic is very wide. We cannot cover everything in one lecture, but still, we will try to learn some basic things. Uh, what does my genome look like? So, if we all have unique genome, right? So, ये ये जो जीनोम है वो हमारा कैसा दिखता है उसके बारे में थोड़ा सा just a minute.
All those points in this video. So, firstly, if I talk about education, so yeah, no need to compromise anything about education because
Yes,
all the
Nikita, can you
parents
please
are
mute
very well
yourself?
experienced, they're very
Nikita,
knowledgeable, they will always
Nikita, please mute yourself. Yes, madam, yes, Shruti ma'am, please unmute yourself.
Yes, Khushbu ma'am. Uh,
Yeah.
Actually, I am facing
Yes.
some problem regarding these tabs. So, okay.
Sharing the screen, right? The screen is not that much visible right now.
Now,
Just do one thing, stop sharing and then share your screen again. Don't do the full screen. In the PowerPoint mode only you can do the presentation. That is okay.
okay, okay.
Yes, now it is visible. So you can change the slide.
Okay,
This will be
this
the
is fine,
title
right?
slide of your presentation.
Fine.
Okay, now it will be the second number slide. So you can do the presentation with this only. That is okay in the PowerPoint mode.
Visible?
Yes, it is. This will be the what is genome, the
Yes,
third
yes.
number slide,
Done,
I
done,
think.
done, done.
Yes.
Okay. So, what is my genome look like? So, if all the DNA from a gene, single human cell will stretched out like we have seen a photo. See this image. In this image, we can see how it's uh, unbinded. We can see, right? So there is a cell, then it is unbinded, which is bounded on chromosome. When we unbound them, it is all made up of so many nucleosomes, histones, and similarly you can see the gene DNA, which is also made up of base pairs. uh, So when we stretched out end to end, it would make a 6 foot long strand comprised of a 6 billion letter code. It's hard to imagine how that much DNA can be packed into a cell's nucleus. This is only about one gene, which is 6 foot long, and we have already so many genes on one chromosome. As a human, we have also 46 chromosomes. So you can imagine the nature, right? The secret lies in the highly structured and tightly packed nature of the genome. What does my genome look like? If all the DNA from a single human cell was stretched out end to end, it would make, okay, this is already covered. DNA double helix. I think this DNA double helix you have already studied in 12th science maybe. So let's cover this faster. Genome are made up of DNA, an extremely large molecule that looks like a long twisted ladder. DNA is read like a code. This code is made up of four types of chemical building blocks, that is adenine, thymine, cytosine, and guanine, abbreviated with letters which we say A, T, C, G. And the order of the letters is this code allow DNA to function in different ways. The code changes slightly from person to the person to help make you who you know. So everyone which are unique to from each other is based on the arrangement and sequence of this ATCG, and that makes us unique from each other. Now chromosomes. Our DNA is located on chromosomes. The DNA in a cell is not a single long molecule. It is divided into a number of segments of uneven lengths. At certain points in the life cycle of a cell, those segments can be tightly packed bundles known as chromosomes. During one stage, the chromosomes appear to be X-shaped. You You can see on in figure also. During one stage, okay, every fungus, plants and animals has a set number of chromosomes. For example, human have 46 chromosomes, which I said already. That is 23 pairs. Rice plants have 24 chromosomes and dogs have 78 chromosomes. Similarly, each and every organism has their own numbers of chromosomes. What causes genetic diseases? A genetic disease is caused by a change in the DNA sequence, which I said already that A, G, C, T, which codes are in sequence in each and every person. That is unique thing. But when something changes in nature, it will convert into some disease. A genetic disease, right, that is caused by change in the DNA sequence. Some diseases are caused by mutations that are inherited from the parents, and our parents are an individual at birth, present in the individual. Other diseases are caused by acquired mutation in the gene or group of genes that occur during a person's lives. So basically, what is genetic disease? That is, genetic diseases are occurred in two ways. One is occurred, one can be occurred that is inherited from parents, like जिसके ऊपर हमारा कोई कंट्रोल नहीं होता है, पेरेंट्स के थ्रू ही वो आता है. एंड सेकंड वन दैट इज एक्वायर्ड म्यूटेशंस. कुछ चेंजेस हुए हमारे जीन्स के अंदर, सो that may lead to genetic disorder or diseases. Genetic variants. What is genetic variants? Changes in the DNA sequence are called genetic variants. The majority of the time, genetic variants at all, but sometimes the effect is harmful. Just one letter missing or changed may result in a damaged protein, extra protein, or no protein at all, with serious consequences for our health. So, only one code or only one missing letter or in sequence that will matter. Additionally, the passing of genetic variants from one generation to the next helps to explain why many diseases run in families, such as in sickle cell disease, cystic fibrosis, and Tay-Sachs disease. If a certain disease runs in our family, doctors say you have a family health history for the condition. And about this, sometime we have already seen about that. कभी ना कभी हमारी फैमिली हिस्ट्री में ऐसे कोई डिसीज होते हैं जो हमने कभी सुने हैं लाइफ में. आजकल डायबिटीज पिक पे है ये. Now, what is genetics testing? Genetic testing consists of the process and techniques used to determine details about your DNA depending on the test. It may reveal some information about your ancestry and the health of you and your family. कंप्लीट इंफॉर्मेशन बता देता है. जब आप डीएनए टेस्टिंग करवाते हो तो आपका, आपकी फैमिली का, आपके जितनी भी आपके पुरानी पीढ़ियां है, सब कुछ आपको डिटेल में बता देते हैं जेनेटिक टेस्टिंग. सो जेनेटिक टेस्टिंग में क्या क्या इंक्लूड है? दैट इज प्रिडिक्टिव टेस्टिंग, डायग्नोस्टिक टेस्टिंग, फार्माकोजेनोमिक्स टेस्टिंग, रिप्रोडक्टिव टेस्टिंग एंड डायरेक्ट टू कंज्यूमर टेस्टिंग. Briefly, फटाफट से ले लेते हैं. That is predictive testing. So what is for what it is done? That is for this who have a family member with a genetic disorder, just like Down syndrome, right? The results help to determine a person's risk of developing a specific disorder being tested for. These tests are done before any symptoms present themselves. Like अगर हमारे फैमिली में कहीं पे ये ऑलरेडी किसी को हुआ है, तो इस टेस्टिंग से हमें पता चलेगा कि ये आगे की नेक्स्ट जनरेशन के अंदर इस जीन्स का प्रेजेंस है कि नहीं. सो इससे हम प्रिडिक्ट कर सकते हैं पहले से ही कि इस नेक्स्ट जनरेशन में आएगा कि नहीं. Diagnostic testing. It is used to confirm or rule out the suspected genetic disorder. The results of diagnostic test may help you make choices about how to treat or manage your health. This testing is mainly um, done in nowadays in pregnant ladies. So अगर जब बेबी वॉम्बो में है उस टाइम पे अगर उसको he will have this syndrome or not. Next, pharmacogenomics testing tells you about how you will react to certain medications. फार्मेकोलॉजिकल स्टडीज में भी ये इम्पोर्टेंट है लाइक अगर हमने कोई प्लांट फॉर्मुलेशन भी लिया या फिर कोई केमिकल बेस्ड हमने जो हम रूटीन लाइफ में मेडिसिन लेते हैं तो अगर कोई मेडिसिन का रिसर्च हो रहा है या फिर उसके बारे में अगर हमें पता नहीं है एलर्जेटिक होगी या नहीं होगी तो एडवांस में हमको उससे पता चलता है कि हाउ विल react these medications. Reproductive testing is related to starting or growing your family. It includes the test of biological father, mother to see genetic variants they carry. So the test can help parents and healthcare provider make decision before, during and after pregnancy. फॉर एग्जाम्पल अगर प्रेगनेंसी के टाइम पे पता चल रहा है कि पर्टिकुलर मॉम के वॉम में जो बेबी है दैट इज having some disorders, having some genetic disorder, serious disorder, like Down syndrome again. So, if it is detected before 3 months, a mother or doctor can decide to terminate that pregnancy. Okay. According to the present rule, we cannot continue with the baby having serious genetic disorder. Next, direct to consumer testing that can be completed at home without a healthcare provided by collecting DNA sample and sending to a company. And okay, by this we can share some, analyze our DNA. Eccentric, kinship, lifestyle factors, and potential disease risk also can be identified from this. Next slide. Forensic testing. It is carried out for legal purpose and can be used to identify biological. ये हमने बहुत बार सुना हुआ है, मूवीज में भी बहुत बार हम देख चुके हैं कि अगर कोई केसेस लीगल केसेस है उसमें जरूरत पड़ती है तो डीएनए टेस्टिंग बहुत बार सुना है बेबी आपका है कि नहीं फॉर एग्जांपल लाइक अगर कोई क्राइम हुआ है वहाँ पे कोई ब्लड सैंपल मिला है तो उसमें से हमें पता चल जाता है वो क्राइम कहाँ हुआ है वो किस पर्सन का है and everything. What are the benefits for my health? One way genomic research can benefit you in through the emerging field of precision medicine and specifically characteristic of your genome can help predict how you will react to certain medications, allowing your healthcare provider to choose the appropriate prevention of treatment options for you. Now, how does genomics impact everyday life? As technology advances and we learn more about how the genome works, information about our genomics is quickly becoming a part of our everyday life. How? Emerging technologies give us the ability to read someone's genome sequence. So, having this information can lead to more questions about the genomics means of for ourselves, our family members, and society. This give us everything and each and every information. Impact on society. Whether you realize it or not, many parts of our daily lives are influenced by genomics, information, and technologies. Genomics now provides a powerful lens for use in various areas, including medical decisions, food safety, ancestry, and more. Okay, as I have to take a lecture on genomics, I have given some basic information on genomics to you, but there is something I want to share to you about some new things that is always we already listen somewhere genetics. Here we are talking about genomics, but I think you should know the difference between genetics and genomics, right? What is genomics and what is genetics versus genomics? That is, both play roles in health and disease. Genetics refers to the study of genes and the way that certain traits or conditions are passed down from one generation to another. And genomics describe the study of all of the person's genes. So, in short, if I say when we have to study about a particular a particular gene or which is on particular chromosome, we can choose genetics. And genomics includes the whole genome, right? कोई एक जीन की स्टडी दैट इज जेनेटिक्स, कोई एक या फिर सिलेक्टेड डिजायर्ड, बट जीनोमिक्स पूरा जीनोम कवर कर लेता है. Differences: genetics involves scientific studies of genes and their effects. Genes, which is units of hereditary, carry the instruction for making proteins which direct activities of cell and functions of the body. But what about genomics? It helps research discover ways some people get sick from certain infections, environmental factors, and behaviors while others do not. It is important for understanding genetics is important for understanding more about diseases caused by a single gene. That is, using genetics and complex disease caused by multiple genes. But genomics is important for understanding more about diseases caused by environmental factors. Can lead to earlier diagnosis, interventions, and targeted treatments. What are some of the new genetic and genomic techniques? That are proteomics, pharmacogenetics, pharmacogenomics, stem cell therapy, and cloning. Here there are two techniques which in we are interested. I should go further. That is pharmacogenetics and pharmacogenomics. See, here also there are both terms used differently. Pharmacogenetics and pharmacogenomics. Pharmaco we know, but there are also difference in pharmacogenetics and pharmacogenomics. Pharmacogenetics is a field of study dealing with the variability of responsibilities to medications due to variation in single gene. So this you have to fit in your mind.
Excuse me, Shruti ma'am.
Yes.
Just a second. Our meeting will be going to end up in a 1 minute.
Okay.
So because this is the free Zoom, so we have to join again after 1 minute.
Okay.
So I just request to, before starting this pharmacogenetics explanation, just we are going to end this meeting and we have to again join in the same link, whichever I have sent you, so that we can get again 40 minutes. So our meeting will be end up right now. And ma'am, one more thing, whenever you are going to end this meeting as your host, the Zoom will be, recording will be stopped and it will be saved in your computer document folder. And there will be a pop-up occurs in your laptop, that will be the recording is converting. So please don't cross up on this, just make this recording convert up till 100%. then automatically it will be going to saved in your computer. Okay, document માં એક automatic folder બની જશે Zoom નું. એમાં તમારું ટોપિક સાથે ડોક્ટર શ્રુતિ ભાવસાર એવી રીતે સેવ થઈ જશે. પણ જેવું આપણે આ મીટિંગ એન્ડ કરીએ ને એટલે એમાં એવું લખાઈને આવશે કે Zoom recording is converting. તો ઈ તમે ભૂલથી બંધ Yes, ma'am, you can share your presentation. Yes, Shruti ma'am.
Khushbu ma'am, my slide is visible or not?
Screen is already shared, but you just click on the slide, so you can directly click on the 10th or 11th number slide, so I can able to say. Yes, okay. Yes, ma'am, it is visible now. You can start. Yes,
Okay,
yes.
fine. So, pharmacogenetics and pharmacogenomics डिफरेंस हमने देखा था, तो दोनों में कब हमें क्या यूज करना है वो डिसाइड करना सबसे पहले जरूरी है. Which stream we should decide for which purpose. So firstly, what is pharmacogenetics? It's a field of study dealing with the variability of responses to medications due to variation in single gene. Pharmacogenetics takes into account a person's genetic information regarding specific drug receptors and how drugs are transported and metabolized by the body. The goal of pharmacogenetics is to create an individual drug therapy which allow for the base choice and dose of drugs. Whereas pharmacogenomics is similar to pharmacogenetics, except that it typically involves a search for variation in multiple genes that are associated with variability in drug response. Since pharmacogenomics is one of the large-scale omic technologies, it can examine the entirety of the genome. Rather than just single genes, pharmacogenomic studies may also examine genetic variations among large groups of peoples. So Basically, it is wide range research, and if we study whole genome, we should choose pharmacogenomics. And if we know the targeted effect on particular genome, we should choose pharmacogenetics. Now, there is a new term I have to introduce to you, that is herb genomics. That is a field of research that combines high throughput omics tools to understand the molecular mechanism underlying the mechanism properties of herbs, which could be applied in both genetic, bioengineering, and medicinal plants breeding. This branch of research is mainly works for the genome research of a particular plant. So currently the usage of plant medicine, now let me take you towards this term. Currently, the usage of plant medicines is regarded as an alternative to conventional medicine, as we know. So, with plant extract commonly employed as part of traditional therapies. To transform herbal materials into botanical drugs, several factors must be considered, including the identification of the plant species, selection, extraction, purification of active molecules, in vitro and in vivo bioassays, quality control, non-clinical safety assessment and clinical efficiency. इन सारे steps में से हम जब particular drug या plant को pass करेंगे, उसके बाद ही एक safety assurance हम clear कर पाएंगे और उसको as a herbal medicine we can consider. Consequently, plant-derived natural therapy requires documented scientific evidence with statistical significance, which leading not only to the discovery of new medicinal compounds but also to a deeper and more comprehensive understanding of plant biology, physiology, and taxonomy. So, genomics, along with its related studies, plays a crucial role in identifying genes involved in plant specialized metabolism. Consequently, traditional herbal medicine has entered a new era known as herb genomics or herbal genomics. This new discipline integrates various omics fields such as genomics, transcriptomics, epigenomics, proteomics, metabolomics, and bioinformatics, offering powerful tools for understanding, controlling the genetic and biological aspect of health. Applications. The emergence of plantomics has provided new insight into the biology and evolutionary history of plant genomes. So it is facilitated the identification of genes involved in the biosynthesis of medically important active molecules and has enabled the discovery of new pathways. Plant genomics has allowed for the genome-based taxonomy classification of closely related species. So, uh, you have already seen that there are so many species for particular genus, particular plant. So when we have to study or discover some species of particular plant, we can choose genomics, plant genomics. Through the plant genomics, we can specify or we can discover or we can give some differences related species. And if we want to study in particular species and for particular genome, we should choose the pharmacogenetics. We should study that. Additionally, it briefly explored the potential applications and key opportunities offered by the omics perspective in the pharmaceutical industry and the agrobiotechnology field. This knowledge can be leveraged in the discovery of plant-based drugs. Now, Khushbu ma'am, this content I have choose to give you some basics regarding this omics, that is genomics. So now I expect from you all what you want. This now this is done from my side. So you have
Yes, ma'am.
you can ask if you have any queries, all of you, or any questions.
Okay, ma'am. You have already covered up a very basic information all about genetics, genomics. So, from the participants, if you want to know anything more about genomics or genetics or related to this topic, then you can ask to our ma'am. If anybody wants to ask,
I
ma'am,
can
can you please do the one thing that just make me host? Okay, I'll do. I'll do. No, I'll do. Don't worry.
okay. Okay.
Yes, anybody wants to ask anything to ma'am? Okay, so thank you so much, ma'am, for giving us a very good information related to the genomics and genetics, because in our pharmacy curriculum, there will be not any topic related to genomics. So thank you so much
Yes,
for
this is
covering and
yeah.
acknowledge us about the new topic. And this is the certificate which we want to provide you as a token of gratitude.
Yes.
So thank you so much. Please accept this.
Yes.
And really, really thankful, ma'am, for giving us a beautiful lecture on the introduction to genomics. Thank you
Thank you
so
so
much.
much to you also to give me this opportunity. Actually, this topic is very wide. We cannot cover this topic in 1 hour, but still,
Yes, yes.
we hope we'll meet again. Okay.
Sure,
Thank you
ma'am.
so
Sure,
much.
yes. Okay. Thank you. Thank you so much, ma'am, once again.
Okay.
Okay. So, everyone, if you want, you can leave the meeting now. Thank you, ma'am, once again. Thank you so much.
Okay.